Well, I'd love to if I actually knew. So I think the fact is that we are collecting all this data prospectively or longitudinally, okay? And just the way we work is that we, from a funding, no, very honestly, from a funding point of view, we weren't doing it in real time, and we haven't, okay? We've collected all the data and we are working on it. And I'm very lucky to work in a big team with a research team, and I've got PhD, fellows, who are working as part of their PhD program. So we are doing it, and I don't think I could actually really. I can honestly say, if I said anything, I'd be wrong because it's too small numbers, and we've only just obviously, as a late breaking, kind of got the PAF-CR rates, the major pathological response rate, and even then, it's still 32 patients, not more. And so the idea is that we will get some really interesting signal. And I suppose maybe one of the most interesting signals, which was discussed a lot, was about the circulating tumor DNA, because it's quite binary and it's objective and it's understandable both to doctors, patients, and scientists, right? So if you have a high ctDNA pre-cycle 1 and you drop it down to 0 pre-cycle 2, does that mean that you are going to correlate to a path complete response? And that's interesting. And we'll get that data, but it's not just the ctDNA, because it's a binary yes, no, or a little bit higher, a bit lower. And that will make good clinical judgments about what you do maybe in the future. But, in order to make those ctDNA, you have to do what we call whole exome or whole genome. And then there's the exciting bit. You can do kind of, much bigger detailed analysis of the biology of the cancer. And then maybe apply that not just to the individual patient, but as a group to subgroups of patients in the future, and help design the next biology-driven trial. So I think it's really exciting. We're showing lots of different bits: microbiome, TCR complexity, and kind of liquid immunobiomarkers. But I think it's a very exciting space to be in. And I've always said that, what I set this up because I knew it'd be a relatively small trial at the beginning, is that learn from every patient. So each patient is an individual, and we get as much information of it from each one, and you can learn more. And we were very lucky because we got funding to expand the trial. So we are going to get another, 40, 50 patients. And then that's really interesting, not just because it's good for the patients, right? And it's good for the science, and I think probably, although we'll get some early readout of the first 32 about some biomarkers, which I really hope that some of it could be presented, if not in the next 3 months, certainly within the next year. And that will help us then build on the translational work for the expanded group. So I think first of all, we've got to expand the numbers so that so there's kind of the primary endpoints, which is obviously PAF-CR was the initial primary endpoint for a phase 2 non-randomized trial. We think that we worked out statistics that you need about 80 patients, hopefully all TMD medium or high, and a certain degree not to relapse to get to what we call 3-year relapse-free survival. And that's meaningful for many reasons, not just for the patients, but also for regulatory. If we are going to go regulatory, to be honest, you're going to say, how many patients relapsed if they had standard of care surgery and chemotherapy? So that's one aspect. If you then correlate that with ctDNA testing, so a lot of people said, look, you've really pushed, like a lot of trials, into the neoadjuvant space. You're only giving immunotherapy pre-op and you're not giving it post-op. Is that the right thing to do? First of all, are there a group of patients who should have adjuvant immunotherapy or different chemotherapy because they've got residual ctDNA? So that's clinical judgment. The other thing is that there's a whole avoidance of surgery or non-operative management. Can some patients who are frailer outside the trial avoid having surgery if they just have a few cycles of immunotherapy? And we've seen that before because we have treated some of our advanced metastatic patients, and we noticed the primary has disappeared, as has everything else, and they don't seem to relapse. So that's a clinical bit. The ctDNA, as I said, is linked in with microbiome. So I must admit, the one thing I really like about this study is that I wasn't really sure about microbiome. I mean, there's a lot of stuff written about it, and now it's all about young onset bowel cancer and things like that. And we just thought, let's go for this. Let's just collect oral samples, swabs, and see. And I would say, because I'm learning all the time, there are some very interesting bacterial species at the baseline. I think they change. I've seen that they change. Whether it's actionable, that means it's therapeutic, is a different issue. And there's whole thing about fecal matter transplants and all this, I don't know. So microbiome is interesting because I don't know where it's going to go, but I think, unless I'm wrong, this would be one of the first trials to actually show whether microbiome does change from baseline to treatment response. The microbiome is one, and I do think this TCR complexity, these kind of neoantigen signatures, they are being done, and I do think that maybe beyond blood itself and the ctDNA and this kind of tumor tissue, yeah, it's a dynamic process. And I do think in the future we might be using kind of these liquid biomarkers, including immune response biomarkers, that will tell you, whether you're already responding, not only pre-op, but also in the post-op phase. This idea that as long as you have immunotherapy downstage and you have oncomprehensive surgery, your immunity is still upregulated. Okay. On the other hand, there's a safety issue. So it came at discussion, what about these toxicities people are having? We do sometimes see people have florid immune toxicities, myasthenia, myocarditis, pneumonitis, and maybe there is something in the blood, that can predict that some patients will get quite severe side effects. And again, that may be also a kind of an unexplained or something that I will find out, which we would have never known. So I think there's so many exciting things that can be done, and will be done. I'm really thankful for kind of doing this study, and it's really a team effort. It takes a lot of people to kind of put the study together, and of course I have to thank the patients, because if we didn't have the patients, we wouldn't do the study. But it does make us think, that look, all these interesting studies are investigator-led studies, non-randomized, everyone using a different drug, duration of treatment, and I think we talked about it after, like harmonization and trying to say, okay, should we all try and work together to a degree, and rather than do all these kind of smaller studies, which is fine at the beginning, is that you start harmonizing. Okay, should we actually have a standard, almost playbook, or saying, okay, if we're going to do these studies, shall we combine forces and say, okay, we all should agree that maybe it's going to be 3 months of immunotherapy. I'm not saying it should be, but 3 months of immunotherapy is the gold standard for generally, and then you can do whatever you like in the experimental arms. But it's an exciting field, and I think neoadjuvant space, not just immunotherapy, generally pre-op space, is a very good place to be, because you can study biology, you get these biomarkers, surrogate biomarkers, much more quickly. And desperately we need them, because a lot of treatments we give in cancer, even immunotherapy, is not really targeted. I mean, they say it's targeted treatment, it's not really. It's just boosting up the immune system for some way that we can kill it. So I think if we do that, then we'll learn more about good treatments, the best treatments for our patients, make them live longer with good quality of life, but also understand cancer and how we can tackle cancer as well.